Good evening, everyone, and welcome to the Public Science Forum, Roche Public Science Forum for 2023. My name is Tika Hayward. I'm the chair of the Roche Science Week Committee for this year and also the lab supervisor for the Bermuda College Science Department. I'd like to first acknowledge the presence of our president. I think she said so just for a minute. Again, Dr. Brady, our president. I'd also just like to say it's really wonderful to see so many of you here this evening. We've been virtual for the last 2 years, totally virtual. So it's really nice to see so many of you come out and support and also be able to include those who are viewing online. At this time, it's my pleasure to introduce Ms. Denasia Curtis. She's one of our 2023 Roche Science Merit Scholars, and she will introduce our guest speaker for the evening. So Ms. Denasia Curtis is a talented 19-year-old currently pursuing her Associates in Science at Bermuda College, having previously attended the Barclay Institute, T.N. Taylor Middle School, and the Gilbert Institute. Denasia has received awards and recognition in social studies, for academic diligence, and for maintaining high honors throughout her high school career. In the future, Denasia would like to become a forensic investigator or a historian. Please welcome Ms. Denasia Curtis, one of our Roche Science Merit Scholars 2023.
Dr. Eve De Rosa is the Mibs Martin Follet Professor in Human Ecology and the current Dean of Faculty at Cornell University. She attended Henry Bay Primary School from Primary 1 to 3 and Bermuda High School for Girls from Primary 4 to the senior year program of the Salters Grammar School. She left the island for Vassar College, where she earned a bachelor's degree in biology psychology. Her Ph.D. training was in animal neuroscience at Harvard University, followed by a postdoctoral training fellowship at Stanford University School of Medicine in human neuroscience. She started her career as a psychology professor at the University of Toronto, and then a decade later was recruited to Cornell University. In her research, she studies how brains and cognitive abilities, such as attention and learning, change throughout the lifespan using a cross-species approach. Her research is currently supported by the National Institute on Aging, and she has served as a member of the Neuroscience Studies Section at the National Institute on Aging, as well as a member of the Recruitment Accelerator for Increasing Diversity in Aging Research at Upstate Medical University in New York City. She is a co-founder of a neuroscience program for elementary school children in Syracuse, New York, a city with a diverse population. This Fascinating Brain program supports our outlook that academia requires a diversity of ideas and a diversity of individuals pursuing them. Please join me in welcoming Dr. De Rosa to the podium.
Thank you, David. Thank you, Bermuda College, for inviting me back home. It's wonderful to be here. It's wonderful to see so many familiar faces. And yeah, welcome, and I'll try my best. So this is my magic drink for the night. So I would first want to acknowledge beyond the generosity of Bermuda College. I'd also want to thank the Roche Holdings for sponsoring Science Week. It's been a blast. On Tuesday, I worked with middle schoolers and then high schoolers, and then tonight is the public lecture. So it's really been wonderful connecting. And I'd like to also thank the National Institute on Aging for sponsoring the work. And this is going to be hard. I fully expected that when I would be giving this lecture, my entourage would be watching online or we got out of the hospital and unfortunately we lost her before I could get home. And so I'd like to dedicate this to her. The elephant in the room. The elephant in the room, and it's funny how I've been interacting with people for the last few days, and everyone has a connection to dementia, Alzheimer's. It's like we all have family stories about cancer. And we have an aging population, rapidly aging population, so this is an urgent thing for us to address. And I think if I was to have a take-home message, really it is not everyone who ages becomes demented, and so that's a good thing. And that's will I have dementia? No, not everyone will. And I guess I'll keep going and I'll keep expanding on that. I think it's important to know that healthy aging does come with changes in the brain and changes in how we attend to the world, learn about the world, and interact with the world. Oh, no need to apologize. Good evening, everyone. And so yes, there are changes in how quickly we can think about things and multitasking, but don't forget that there are things that stay stable and stay the same. And maybe we get better. In our lab, we actually create things where older adults outclass our young adults on some of our tasks. And so we don't, oh my God. So you don't want to forget that there's so much wisdom that gets passed along with age and expertise. And so it's normal to occasionally forget your keys or pay a bill. And so what we want to differentiate is healthy aging from pathological aging. I focus on healthy aging, so I am not a medical doctor, and I don't study Alzheimer's disease, but what I'm trying to do in the lab is to get in there early. Can we start to look at ages 40 and 50 and see if there are indices that are telling us that someone's at risk for dementia? Because right now, there isn't a cure. All we can do is intervene early and slow or delay the progress. And so we're just trying to find different ways using the body and not just the brain to predict that someone's at risk. So I found this little infographic. I found it's helpful to know that in healthy aging, yes, sure, we'll forget things, or sometimes we'll not know the word that we want to say or what the person's name is, but the pathology, when it becomes something pathological, when you forget people you know deeply, like your spouse, like your children, that's when we're talking about dementia. When you can no longer manage your bills. And so it's when it interferes with your daily living. And these are just infographics you can find on Alzheimer's Association and all this CDC. They have lots of different infographics for you to know the difference between what is healthy aging and what's pathological. And I think a lot of people have anxiety that's unfounded. And so what I want everyone to do right now, take a deep breath and let it out. Okay, you're gonna do it. And so this idea of interfering with daily life, that's when we're getting to something pathological. But what you don't always hear about is that 40% of people who might be at risk can stave off the pathological dementia just through healthy living. And so that's what we're going to feature. And so we all know what to do, there's no doubt. We've been told to eat well, exercise, stay connected with our families and friends, sleep well, all of those things, if you do them, diminish your risk for dementia or Alzheimer's disease. And so what we want to do now in the lab is try to understand why, because if we can understand why, that gives us mechanisms to intervene. And maybe eventually to maybe cure. Unfortunately, I had to switch machines to a computer, so you won't get to hear this very adorable 4-year-old ask me the important part of, is the brain the most important part of the brain? I think he's expecting me to say yes, because he knows I study the brain. But it's complicated. So those nerves from the brain go throughout the body. And one of the nerves that I'll be talking about today, the vagus nerve, actually is like the sentinel, the wandering nerve that goes throughout the body, takes in all of the sensory information from the world and sends it back up to the brain. And importantly, the vagus nerve is like a superhighway from the brain to the heart. And so what we're going to try to do is understand the behavior of the heart, and can that be a predictor of what's happening in the brain? Oh, no audio, but it's adorable. And so this is just to give you an index. So when we're showing, this is called the mid-sagittal section right here. And basically that's if you split the brain in half, you see the inside. And of course, this helps you to see what's the front of the brain and what's the back of the brain. Most times when people talk about the brain, they talk about these very high-level cognitive things that we do, setting goals, emotions, memories, attention, learning. So a lot of that is happening here. But I think what's underappreciated is the bridge between the brain and the body. So this is where we're going to be spending our time today. That is where the plaques and tangles that probably all of you have heard about, they start there. They start in your 20s. Like all of us are accumulating them. And it's one of the things we know why sleep is important. When you sleep, it's restorative, and it actually helps clear some of the plaques and tangles. And so sleep is a really important part of your mental cognitive health. But this brain-body connection here also is where all the neurochemicals get created. And the neurochemical that I've been studying since graduate school is called acetylcholine. And when someone's diagnosed with Alzheimer's disease or mild cognitive impairment, which is kind of like a precursor to Alzheimer's disease, they intervene with this neurochemical to boost its function in the brain. And it turns out that that chemical doesn't cure Alzheimer's disease, but it does treat the cognitive decline and slow it down from progressing too fast. But there is no cure yet. So the NIH, the current project that we're working on with NIH, is looking at the heart and the mind and how they're connected with this neurochemical. And so we want to know here, the vagus nerve, that superhighway between the brain and the heart, whether that is a place that supports healthy aging. And then we're asking specifically, is this going to be a place where we can intervene to help people? And then here, the neurochemical that I'm studying, acetylcholine, is actually what is allowing for that communication between the heart and the mind. And so can we then also understand the neurochemistry of this and for development of pharmaceuticals, hopefully. And then lastly, we're developing technology so that people can eventually, if we're right, and we can, we support our hypotheses. We know that healthy cardiac behavior and social connections are all good for healthy aging. And so can people share their heart behavior with each other? And we even did a little prototype with my mom. Her sister is very lonely, and so my mom contacts her, and they do deep breathing together. So this idea of social connectedness while doing healthy behaviors with yourself. Time to nerd out. Okay. The acetylcholine is at the heart, it's in the brain, so we know it supports cognition in the brain, it helps us learn and remember things. We know it declines and is associated with decreases in learning and memory. But in the early 1900s, they didn't know that there were neurochemicals in the brain. And so this was the first experiment to let us know that. And so What they had was a heart with the vagus nerve connected to it. They put it in a petri dish with solution, and they stimulated, and the heart slowed down. So that they knew that. But what they didn't know is that what was being released when you stimulate the vagus nerve could be used to change the behavior of the heart. So here we have the heart with no vagus nerve, right here. And what they did is they just took the solution from here and squirted it over here. So heart B had no vagus nerve, but as soon as they squirted what was around surrounding the heart area, it caused the heart to slow down. So that was the first index of, or evidence of, chemicals that could then act along the nerves to change the behavior of an organ. And so that is acetylcholine, and this is the neurochemical we're talking about today. The other thing about acetylcholine is that it changes across the lifespan. And so just like we see the sun rising here, and then at the peak of the day, and then sun setting at the end. When you look at enzymes that support the synthesis of acetylcholine, you can see these are decades down here, so 10 years old, 20, 30, 40, 50. We're kind of at our prime at 40, and then it starts to decline. And this is just normal aging. As I mentioned, this is just what happens. And there's nothing wrong with that. What's wrong with that are these people right here. Those are people who have Alzheimer's disease, and they are showing a pathological reduction. And at age 60, you can see this is where they should be, but they're down here. And so this is pathological aging, and this is why this data suggested acetylcholine was the way to intervene. And so we talked about the fact that we know what to do, but let's get on to why. This is going to be sort of a leap. Take this leap with me. So, acetylcholine and the vagus are important to this rest and digest. So, how many of you have heard about fight and flight? Right? We've heard about that? That is the sympathetic system. That's when you are under threat, and your sympathetic system then goes into overload. You don't want that chronically. You will kill cells in your brain for sure if you are under chronic stress and you're driving that system. It's fine to have a threat, it's fine to have that, but you must then put on the brakes. That's the parasympathetic rest and digest system. And so that's the system that we're going to investigate for whether that's where we can intervene. It has acetylcholine, it has the vagus nerve, and we think that's restorative. And so that's where we're going to spend our time. I'm going to show you some rat data. I'm going to show you some human aged human data, and it's weird to say like humans, they have to reference rats. And then I'm going to show you an intervention that taps into the vagus nerve. And so we're going to study cognitive flexibility. And so what I mean by cognitive flexibility is being able to adapt to your surroundings and tending to the right thing that's important to the goals you have. And so we'll start with that. I want to let you know that not many people do this, but in our lab we do both human and rat data. And the reason we do that is, one, we're allowed to, because when you look at the humans and the animals, they have what they call homologous or similar neurochemical systems. And so in the rats, we can be a little bit more invasive. We can manipulate the chemistry of the brain cells directly. We can record more directly from those cells, unlike humans. And then with our human participants, we put them into the scanner and we do what is called functional magnetic resonance imaging, so fMRI. And that allows us to look at blood flow. So when somebody's doing a cognitive task in the scanner, we can see what parts of the brain are important to them achieving the outcome, the behavioral outcome we're looking for. I just want to share with you, just simply when we're learning something and we want to store it and then remember it, there's always this competition between the thing that's coming into our brains that we're supposed to be learning and things that we already know. And what we want a healthy brain is to interweave the good things that we want to learn right now for the goal in hand, and we want to suppress those things that no longer are relevant to what the goal is at this point. But there's always this competition between bringing the information in and learning it, or remembering it and recalling the things we already know. And so, what acetylcholine does in learning, it allows you to encode and learn the thing that's important to your behavior right now. So you walk into a new learning situation, your brain is flooded with acetylcholine. But when you're just remembering things from the past, it's not. And so that's a really important thing. We want to enhance the thing we're trying to learn. The other thing acetylcholine does, it suppresses past memories. So any connection between neurons that was strengthened before is suppressed. So it's doing both of these things. It's enhancing what's in the present and suppressing the past. And so that allows the signal to be stronger. And then this is just showing a little cartoon of some brain cells. So if we had two brain cells and they had strengthened a connection between them, they learned something that, when this is together and this is together, I do this, and that's been strengthened. What acetylcholine does is suppress the past and enhances the present. So this is the information coming into the system to be learned. Without acetylcholine, then the memories dominate. And those of you who have lived with someone with dementia will know that they're living the past. They're not here with us at this time. So my grandfather passed away from Alzheimer's disease. He remembered my grandmother, he remembered my father, but my sisters and I were the nice ladies. And so he also thought at that time that he was in the era of Eisenhower. And so you can imagine if you're letting those past memories dominate and that's the space you're in, it's hard to bring in the present. And so that's what acetylcholine is helping us do. And so here, those past memories tend to dominate, and you can't get in that new information. Now imagine, and this was the test, imagine when things are really highly related. So I have this problem all the time. So if you meet somebody at a party and you get the wrong name, for me, that is your name. I don't care how many times you correct me, that is your name. And so that's called proactive interference. And so I give an example here that I thought her name was Julie, but it's really Judy, and I never got it. We call that interference, where what you learned in the past interferes with your ability to learn in the present. And so proactive interference, you imagine if you know Spanish, and now you decided to do Duolingo, and now you're going to learn French, those Spanish words are going to dominate until you can suppress the Spanish and learn the French. And many of us have gone through many keyboard switches, and our fingers are doing all random things. So just want to give you a sense of what it feels like for proactive interference. Audience participation time. Okay, so 2 colors in here, red and aqua. I don't know if you're going to pass this test. But this is actually what we have the rats do and the humans do. So we need our tasks to be simple because rats are not verbal. And we need them to be able to tell us what they know. So we have our humans and rats do the same thing. So we use colors with humans, but odors with rats. And so, what side is red on? Yell it out, I want to hear it. Confidence. Thank you. Aqua. And I helped you. I even told you what your targets are. So that is just, you just do that. We present those every 650 milliseconds here. And we don't tell you what your targets are because you're going to be like rats. You actually have to figure out the rules. So you didn't go to red, you're going to hear a eh. But when you hit red by chance, you hear ooh, ding. And then that's the thing that keeps you going and you start to figure out, oh, that's what they want me to do. Just like the rats, you figure out the rule. They're sneaky psychologists. So, now your targets are green and pink. Where's green? Christine? Thank you. As you can see, this is the sneaky part. This was paired with something you knew in the past. I just told you to tell me where red is. You learned it really, really well. And now I say, just tell me where green is. And you go to red because that's what you learned. You have to unlearn the past in order to learn the present. And that's all we're doing. We're just watching people make that transfer from what they knew before to what I want you to know now. And then we just, because we really want to understand what's happening in the brain, we have to have the right experimental controls. And so this is just a very simple control. What does the brain do, or what do people do, or what do rats do when they just have to learn something new that has nothing to do with their past? So we have the experimental condition where there's a conflict between what you know and what you're supposed to learn, and then something where you can just learn something new. And we need that just in order to interpret how hard is it just to learn something new versus how hard is it to learn something that conflicts with what you know. This is an example with rats. Banana and cinnamon, tell us where banana is. We'll clove and banana together. Don't tell me where banana is, now tell me where clove is. And then where lemon and almond, tell me where lemon is. So lemon has nothing to do with their past. It's a very lovely smelling experiment. And if you're a rat, and we built apparatus for them, so they're water restricted, so they're thirsty. That motivates them to do the job. Odors come out of these ports. They have infrared beams across there, and they break these infrared beams and they can tell us which of the odors is the one that's going back on water. So just like you did, they do the same. I'm just going to show you this behavioral task in young and aged rats. Let's try that again. There, young and aged rats. We're going to manipulate acetylcholine just in a very simple way. Just remember, acetylcholine is the neurochemical that connects the heart and the brain. It's the one that they intervene with the Alzheimer's disease. This is something that supports our ability to learn new things efficiently. And then we're also going to just show you some recordings from different areas of the brain that are important to attention and learning. And so again, when A and B are together, tell me where A is. When C and A are together, tell me where C is, not A, and then D. And so we have our rats learn A, B, and then we have to learn the proactive interference or the novel stimulus. And this is a drug, scopolamine. You probably had it this is what they use for seasickness. We know also when chemical warfare happens, they have people take a blocker for this because they overdose you on the acetylcholine through chemical warfare. And we found out in Israeli soldiers who chronically take scopolamine, they don't have PTSD because they're not encoding the new memories at war. So it's a very meaningful drug. And so we just do this in the rats, block their acetylcholine. It blocks it in the brain and the body, because acetylcholine members at the heart, it's also at the muscles. And then this drug right here doesn't pass to the brain, it doesn't pass the blood-brain barrier. So now we have that experimental control where we know what blocking acetylcholine in the body does versus the brain. And so each rat received each drug across different odor sets. This is our nice young rats. Rats live to about 2 years old. So these guys were, adults. And I just want to show you the 3 different conditions that we talked about. So this is just accuracy. These are the number of days that we tested them on the task that I described. This red line is the A, B, go to A, that first thing that they learn really, 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 really well. And then the blue line and the green line are the new things that we want them to learn. This is the new thing that interferes with their past versus the thing that doesn't. You can see they acquire the blue line much better because they don't have to deal with the interference from their past memories. When we give them scopolamine, they start to not be able to acquire the proactive interference. They keep going to their past memories and never learn the new memory. So is that okay? Is everyone good with that? And interrupt me anytime, I don't mind, I promise. But you can see a naturally aged rat right here under the control condition where we're just blocking acetylcholine in the body and not in the brain. They look like a young rat with scopolamine where we're blocking acetylcholine in the brain. So this gave us our first hint that acetylcholine is playing an important role in the ability to learn this new information that interferes with their past. And so these rats are living in the past, is what that tells us. They weren't able to come and learn the new thing. And then also, when we blocked acetylcholine in the brain, they still couldn't, I thought they would get worse, but they didn't. And that suggests to me that they don't even have acetylcholine available to them to be able to change anything. And that's why when we intervene with pharmaceuticals with Alzheimer's disease, you want to get in there early, while the system actually can benefit from the boost. At this point, they can't. And these guys are 21-ish months, and they will only live till 24, so they're quite good. This is just showing that we recorded between the electrical activity in the behaving rats. I think that's enough for us to know here. And what I would just point to is that this is looking at when we have the past memory interfering with their ability to learn. What we find is in our young rats, they had a nice strong signal here, and that correlated with them being able to learn the new thing that interfered with the past. But when we gave them scopolamine, that disappeared, and they were no longer able to learn the new thing, and they let those past memories dominate. That looks awfully like our older rats when we're not blocking acetylcholine, just naturally what they look like. We also show that here, this is the scopolamine, we're showing the likelihood of being correct. And you can see that on the PI trials, which is the interference trials, the likelihood of being correct is correlated with how much brain activity that we saw before. And so that's just showing the relationship that signal that we saw in recording from their brains is meaningful. It actually predicts their behavior and their ability to learn. This is my sort of first attempt at intervention, and basically, can we reverse the effects of age on this change in our ability to do this cognitive flexibility task? As you remember, we have our superhighway, acetylcholine going along here, and here's the brain and here's the heart. And we have older adults and younger adults doing this proactive interference task. And this is our control task this time. And basically, that is when A and B are together, tell me where A is, which that was their past memory. And then just flip it. When A and B are together, tell me where B is, not A. So it's a little different. It's a subtle difference, but a meaningful one, because people can switch. You get the exact same stimuli again, people switch quite quickly. They have a little problem, and then they switch. But when we ask them to do a new thing, then it's a little bit more difficult. And we also have here, our human subjects are just doing deep breathing. And having 6 breaths a minute stimulates the vagus. And this is actually something that can strengthen what we call vagal tone. And it can also help that rest and digest system, that parasympathetic nervous system. It can promote a strength there so that we put on the brakes on the fight and the flight system. And this is where we think we can have do good. An important experimental control as well is we had people looking at a tone on the screen telling them when to breathe in, sort of like we did before. Breathe in, breathe out. And we set the tone so that it's at 6 times a minute. We also had them do it 17 times a minute because that's our natural breathing state. And so basically that allows us to subtract out the fact that you're paying attention to a tone and all these other parts of the experiment that we don't think are going to be meaningful. So this is our parasympathetic Acetylcholine is going to help us with the rest and digest. What it also does is when we stimulate the parasympathetic system, remember we slow down the heart, right? We stimulate the vagus, it turns on the heart, and it's doing that with acetylcholine. Deep breathing does that. And the thing that we measure to tell us how well we're stimulating the vagus is we look at heart rate variability. And what heart rate variability is, and this is not to do anything to do with like the health of your heart. So I'm not a cardiologist, but this is an index where when you have a heartbeat and you see a cartoon, they always give you that stereotype of like metronome or something like that, something predictable. If you have a predictable heartbeat, you have poor health outcomes. So you will find it with people who are close to mortality, or if they're dealing with cancer, you will see that those start to get more like a metronome. And that's what we call low heart rate variability, because between heartbeats, it's very predictable and not very variable. We want high variability, almost like a tennis player that's sort of standing there ready to go anywhere when the ball comes. You want your heart to do that. It's ready for anything that comes at it. And so that's high variability is good. Low variability is bad. And so you can see what I mean by variability is here is 822. Here's 857, not 822. Here is 752. It keeps changing. And the more it keeps changing, the healthier you are and the better your health outcomes. If it was always 822, we have a problem and you should be at the doctor's. So, and you can measure it. There's tons of apps out there that you can just put your finger on your camera, it'll give you your heart rate variability and tell you where you are relative to your age and gender. So it's an easy thing to do. Deep breathing will make that heart rate variability high, and so it's a good thing. I just want to show here, when we correlate somebody's heart rate variability with what's going on in the brain, we see the brain system important to acetylcholine ability to support learning and memory. So that's just what this is showing. I'm not going to go into the details. This is just showing that how many times somebody's breathing per minute, and this is what we call the sham, where your normal 17 breaths a minute versus our deep, slow breathing 6 times a minute. And this is just letting us know that people are following the instructions. We like that. Here is just showing how many times young people did it. They did a good job here. They're hanging around 6, and here they're hanging around 17. Our older adults are also doing extremely well, around 6 and around 17. So this is just a control letting us know that people are following instructions.
Question,
Yes.
so the person who has a fixed rate on a pacemaker,
That is to get the cardiac muscles to coordinate. So when the heart is beating, we want the muscles to work together. We want them to work together here, and then like 10 seconds, or not really that, but let's say that 822, they work together again, and then 700 work together again. So the idea is that you want your muscles of the heart to work together, those cells, but not to be predictable when they do it again. Does that help?
almost.
Okay.
I think of the person who's got a fixed rate. Is that going to impact on the production of the acetyl?
Beautifully done. So what that suggests is that they probably have lower function, this is the adjective for acetylcholine is cholinergic. So they have lower cholinergic function at the heart. And so that just means that what's receiving the acetylcholine is not as efficient. And that's what's happening in the brain as well. Is that helpful? Thank you for asking questions. So this is just showing that when you do the slow breathing, your HRV increases. And this is just also, if you look at the blue, when they do sham breathing, they're around 120. When they're doing deep breathing, they go to about 160. That means it's getting higher. our index of HRV, or heart rate variability. Higher is better. And same with the older adults, they were hanging out over here around 90, and they moved to about 120 with the deep breathing. This is showing how well they did on the interference task. Were they able to pick up the new knowledge that interferes with their past? And so here is our young group. Let's focus on them. This is now them doing the 17 breaths, which is our normal breathing pattern. They had some difficulty with the interference, but then they overcame it pretty quickly. And then also this is our slow breathing, and you can see they don't have that initial dominant past memories. They actually pick it up pretty quickly. This is with sham breathing, and this is with the slow deep breathing. Now our solid line is the older adults. And you can see the older adults have much more difficulty than the young people initially acquiring the new learning that interferes with their past. So the past is dominating and getting less and less dominant, and then you get to young people's performance. So you get there, just not as efficiently. That's fine, you get there. After deep breathing, look at that. Our older adults are looking like the younger adults, pretty close, but then exactly like them.
So if I'm trying to learn a new task, is deep breathing while I'm learning a new task?
Oh, I love you. Did I pay you? Yes. So that's the idea. Like, we're thinking if we can create technology that will have you sort of stimulating and promoting that parasympathetic rest and digest vagal sort of tone while learning something new, while being together. And things like social support stimulates the rest and digest system, is restorative, is good for us. So that's exactly what we're hoping, that bringing those things together while learning new things or, yeah. So we are excited by these data, and we're working to understand it better, but it's a really promising start. And so this is just me reminding you what interference is, and I will never learn Judy's actual name. This is just showing that this signal that disappeared right here when we block acetylcholine in the brain looks a lot like our older, well, actually, I put the wrong data there, but this is important. Scopolamine blocks that brain activity, and that blocking of the brain activity doesn't allow the animals to learn the present, and they stuck in the past. And then the brain data tells us that when activity is in a certain form, it predicts their poor performance. And it looks a lot like our older rats. And then this is our older rats, rats that are young with the blocking of acetylcholine, never learn the new thing and only get stuck in the past. And then with deep breathing, we can have older adults change to look like they were young again. And so cognitive flexibility with our task changes with that aging, natural aging, but that we can intervene either with acetylcholine or with deep breathing to promote this ability to suppress the past and learn in the present. And then I've been looking at the demographics and just trying to understand what policies we have here at Bermuda. And one of our challenges, and this is something that's being addressed, is the fact that we have here a lower birth rate, life expectancy, so people are living for longer. We're an older population. Proportion of seniors is getting higher. And so there's a greater proportion of elderly residents that need to be supported by our working populations. So this is a challenge. I would like to end with an opportunity. And so I actually think Bermuda College is very well situated. We have a very small population relative to other places in the world. We have more racial diversity than other places in the world. And there's 64,000 of us. Bermuda College already has nursing programs. They produce RNs and LPNs, and can we get certification in geriatrics so that we are working with our older populations and getting them the appropriate health care and hopefully neuropsychological care. And then we have Dr. Walden, who's an adjunct professor at the Bermuda College, who's doing genotyping, and I've heard that she wants to genotype the whole island, and I hope she does. That is, nobody has anything like that. Those data would be incredibly valuable, because we do actually have quite a high rate of dementia on the island. And Roche is sponsoring Science Week. Maybe Roche Pharmaceuticals can also do clinical trials in Bermuda. I feel like that's a really good partnership, so I hope that something like that happens. Thank you.